Hello, this is a disc of Cedrela faecilis, a widely distributed species in neotropics, and dendrochronological potential confirmed by many studies. The most common parameter measurement in the rings is the ring width, and recently stable isotopes provided relevant information about climate in Amazon. But what other information can chemical traces in the rings give us? Come in and discover.
Hello, my name is Daiga Ricardo Ortega Rodriguez. I'm a doctoral student of University of Sao Paulo, and today I will present the work Climate Signals in Triggering Chemical Components of Cedrela faecilis growing in Brazilian Amazon. Topics of presentation. I will start with a brief introduction about what is dendrochemistry and the function of some chemical elements in plants. Then I will present the methodology, the study area and sampling, and the technique used in this work, named X-ray fluorescence, focus in the equipment and the configuration of the equipment for this analysis. Then I will present the results about wood chronologies and climate sensitivity of chemical wood traits. And finally, I will talk about the future approach of dendrochemistry for multiproxy analysis of climate in neotropical areas. What is dendrochemistry? All trees capture chemical elements through two pathways, one by deposition and addition of atmospheric particles in the leaf and bark, and another by direct uptake of minerals in the soil solution by the roots. These elements can be deleterious or essential. Deleterious we do not fulfill any physiological function, and essential that are important for the metabolism of tree. Both are allocated and translocated in the xylem formation. These interactions are archived in the wood of trees by the activity of cambium layer and is regulated by genetic expression, function of the element in the tree age, environmental factors, and climate during growing season. So, the annual rings becoming a time capsule on environmental and climatic aspects of the site. Some elements are important for the metabolism of tree and are part of different cells in different structures of the plant. Phosphorus, for example, is stored in the cytosol of newly formed xylem cells. Potassium involved in cell expansion, and cambium is involved in cell differentiation. Both elements are involved in the cambium activity. Potassium that express how higher are the cells, and calcium how many cells produce the cambium activity. Manganese and aluminum blocks the absorption of other elements and are a good indicators of soil change, especially the pH of soils. Sulfur is essential for defining cell architecture. Iron is part of the chlorophyll synthesis, and strontium acts as an analog for calcium, similar to manganese. Study area and sampling. The study area is located in Yamari National Forest in Rondônia, in the southern Amazon basin. In this region is described a strong seasonal variability of South American monsoon system. The wet period goes from September to May, and dry period normally goes from June to August. The region presents positive significant trend in annual temperatures since 1975, with increasingly frequent drought events since 2000. Samples were collected in an area of forest concessionary. These were cut from 20 nitrate fellet, and 4 radial samples from each disc were cut from measurement and considered the ring width chronology. For each radial sample in radial section, 10 radial section was cut, for measurement and construct the wood density chronology. Each thin radial section presents a 1.7 mm thickness. The best 11 trees of both chronologies were selected and wooden slide, also with 1.7 mm thickness, were used for chemical analysis. X-ray fluorescence. 14 radial samples of trees was analyzed by micro X-ray fluorescence in an iTrax multi-scanner system of Laboratory Dendro de la Vide in the University of Pablo de la Vide in Seville, Spain. iTrax was operated at 30 kilovolts and 50 milliamperes with a copper tube, and samples were exposed to the X-ray beam for 10 seconds at each measurement point in radial direction. Each measurement point have 50 micrometers step side. Count rates of fluorescent photons of elements and radiographic grayscale image in each measurement point of each sample were obtained. The radiographical image were cross-dated with previously built chronologies in each site of assigning the element-specific count rates of each annual ring using Windendro. An average annual time series for each radii were produced. Results. For the period of 1901 and 2018, the ring width and ring density chronology based on 69 and 52% of sample trees respectively presented a significant mean correlation among the series expressed by R bar higher than 0.25 and EPS higher than 0.85. The dating of this chronology was verified by radiocarbon analysis. The chemical chronology is constructed with 20% of sample trees present an acceptable mean correlation among the series expressed by R bar higher than 0.2, but the EPS is less than 0.85, except the chronology of potassium. In drier years, narrower and less dense rings present a decreasing concentration of chemical elements such as sulfur, potassium, and calcium, while others increase such as strontium. It is expected since atmospheric change alter the chemical characteristics of the soil and influence the translocation and allocation of elements in the tree. Cedrela officialis grow throughout the rainy season, mainly between September and March, and it prefers warmer conditions at the beginning of growth. The density presents greater correlation with the final period of the rainy season. The location of elements such as aluminum, potassium, sulfur, and calcium are also correlated with the rainy season of the current year as well as the year following the growth period, while warmer conditions do not favor allocation of phosphorus and sulfur, contrary to calcium. Furthermore, the distribution of elements in xylem cells is not random. Sulfur is mainly distributed in parenchyma that forms between the end of the previous rainy season and the beginning of the current rainy season. On the other hand, elements like calcium, potassium, and manganese are mainly distributed in fibers. The responses of wood chemistry to climate change are currently essential to forecast the impacts on soil nutrient availability or imbalance and forest functioning. In this way, future studies in neotropics using dendrochemistry can help us understand how climate change that influences increasingly frequent dry periods alters the allocation of chemical elements and if this alters the physiology and ecology of the trees. Dendrochemistry can also be used for climate reconstruction of seasonal periods that are not recorded by ring width, density, or stable isotopes. On the other hand, specific ecological responses of each species can be analyzed through dendrochemistry, since we have a variety of species with different anatomical characteristics, in addition to different climatic factors that influence these records. So, what other questions can dendrochemistry and multiproxy approach help us answer in the tropics and in South America? Thank you so much for following this presentation, and thanks for all of this institution. And if you have any question, please follow our social media and have a good Congress. Bye bye.